Good morning. Thank you all for joining. I'm Paul Smith from Angle PLC from our Toronto office today. And I have the great pleasure of introducing Professor Nicola Aceto. Professor Aceto is the Swiss National Science Foundation Professor of Oncology, and also the group leader of the Cancer Metastases Laboratory in the Faculty of Medicine at the University of Basel and the University Hospital in Basel. Professor Aceto and his group are well recognized as leaders in CTC biology research, particularly in the area of understanding mechanisms associated with clusters of CTCs, and have published much of the seminal work, both originally out of Massachusetts General Hospital in Boston and more recently out of the Basel laboratory. And Professor Aceto, it's a great pleasure to have you here today, and thank you for taking the time to share your research with this group. Thank you.
Thank you, Paul, for the introduction. It's my pleasure to be here today, and also to give you an update on our research on CTC biology and the implication of our research for the clinics. So let me jump into the presentation. So to make sure that we are all on the same page, I will recap some information that most of you are probably aware with. And the fact that cancer cells from a tumor, pretty much any type of tumor located in, I would say, any location that you can imagine in the human body, some cells within this tumor will have the properties that enable them to leave the primary tumor site, to enter the blood circulation, survive in circulation, and that's where we call them circulating tumor cells, or CTCs, and then extravasate and form a metastasis at a distant site. So this process here is how cancer kills. So the vast majority of cancer types kills because they are able to spread to distant sites and really form almost like a systemic disease, which is extremely hard to eradicate. And my laboratory here in Basel studies the biology of these circulating tumor cells that are literally pioneers of the metastatic process. And the goal of our research, beyond understanding their biology, is to identify new treatment opportunities to suppress the metastatic spread of cancer. So my presentation today will be divided into 4 parts. First, I will tell you a little bit how we capture circulating tumor cells from the blood of patients with cancer and also from mouse models. Later on, I will tell you why in the past few years we decided to focus particularly on clusters of these circulating tumor cells, so on multicellular aggregates of CTCs. Then I will switch to the biology, deep dive into the biology and vulnerabilities of these CTC clusters. And ultimately, I will tell you what is coming up next by means of translating our findings into clinical studies. So CTC capture, I think this is an important topic, and the reason why this is an important topic is because of what you see on this slide. So this is a tube of blood. You can imagine the following situation. We could argue that this tube of blood has been taken from a patient with metastatic disease, and the patient is not responding to any treatment, and the disease is progressing very fast. So really trying to imagine the worst possible scenario. And in this tube of blood, which is approximately 10 ml, you will find 50 billion red blood cells, 50 million white blood cells, and somewhere from 0 to 10 circulating tumor cells. So as you can imagine, the CTC field for many years, it had little to do with biology. It had mostly to do with engineering and with the need to be able to reliably fish out 2 or 3 CTCs from more than 50 billion other cells. So extremely challenging and a classic needle in a haystack situation. I will spare you from a review of all the technologies that are on the market now. I think there are probably way too many. I will directly focus on the technology that we use in my laboratory. What I would like to stress here is that we do not trust markers for isolating circulating tumor cells, at least not to a very high degree. So we prioritize an antigen-independent CTC isolation procedure, and we exploit certain physical properties of CTCs, such as their bigger size compared to blood cells and their lower deformability. So one technology we are using in the lab is Parsortis. And we use it the following way. So we take a blood sample, typically from a patient. This is peripheral blood or from animal models. We, without any manipulation, we run the blood through the microfluidic device. And you can see here an image from or a cartoon from the top. It looks like there is a serpentine. And the cross-section of this serpentine is represented by a narrowing channel which leaves an opening of 6.5 micrometers. So 6.5 micrometer is a magic number because it's the size that allows passage of red and white blood cells because they are smaller than 6.5 micrometers. And at the same time, entrapment of circulating tumor cells because they are bigger than 6.5 micrometers and clearly less deformable than blood cells, so they are not able to squeeze through. And this way, we enrich for circulating tumor cells in an antigen-independent way and virtually from all cancer types. And since a video is probably better than 1,000 words, I would like to show you this in real time. So here we are imaging from the top. I hope the video works well in streaming. So you can see imaging from the top, and you can see blood cells as they pass through the device. And you can see that there is a region here that is darker, and this is the 6.5 micron narrowing channel. Blood cells flow through this channel, but there are some cells at the interface of this narrowing portion that are stuck. And when we turn into fluorescent mode, we can confirm that these are circulating tumor cells. You see them here labeled in green because they express our markers of choice. So this is essentially to tell you that with using dedicated technologies for CTC isolation, we are able to very reliably isolate circulating tumor cells from the blood of patients with multiple cancer types. We developed in my laboratory protocols to achieve a capture rate which is near 98 to 99% of circulating tumor cells in breast cancer. And we apply these protocols to many different cancer types. The reason why we focus on circulating tumor cell clusters is represented in the next couple of slides. So this is a cluster of CTCs from a patient with prostate cancer that we investigated at MGH in Boston. So you can see in green is staining for PSMA. In red is staining for the white blood cell marker CD45. So this particular cluster has also some white blood cells associated to it, and I will tell you a little bit more about these white blood cells later on. But when we found clusters in the blood of patients, we were first of all quite surprised. This was back in Boston, and we essentially didn't know anything about them. So we didn't know whether they were involved in the metastatic process, whether they were actively seeding metastases, or whether they were just a passive entity derived from tumor, from cancerous lesions throughout the body. So we wanted to first of all understand what is their role in the metastatic cascade. The first thing we did was to look at the blood of patients with progressive breast and prostate cancer enrolled at MGH, and we took blood at multiple time points over the course of their disease. And then we enumerated the presence of CTC clusters or single CTCs. And in breast cancer, for example, you can see that the majority of patients has only single CTCs in their blood at the given point in time. But then there is a fraction of patients in whom we could find CTC clusters, and there is a smaller fraction where the number of CTC clusters was higher, and the time points in which we found clusters were greater than 3. And in this particular population, we could see that progression-free survival was very much reduced compared to patients with single CTCs or only occasional CTC clusters. In prostate cancer, it was a little bit different. In prostate cancer, we were able to follow up these patients for a very long period of time, and we could assess overall survival from the time the blood was taken. And we saw something extremely clear. So we saw that whenever these patients had at least one CTC cluster during just one time point in their life, the resulting overall survival was very short compared to patients with only single CTCs. So these experiments here literally got us started on the field of CTC clusters. So we saw in patients that whenever there is a cluster in the peripheral circulation of a cancer patient, this is a bad news. Yet we didn't yet know what are the consequences of that, whether the cluster itself is able to seed metastasis. And going very much fast forward here, because this has been published a while back, so you can freely access it online. We made use of mouse models. We labeled cancer cells with multiple colors, and this way we were able to distinguish CTC clusters from single CTCs, because CTC clusters were multicolor and single CTCs were single color, and their contribution to metastasis respectively. So you can see a multicolor metastasis here derived from a CTC cluster, and the single color metastasis derived from single CTCs. So this model here, and this is just a control experiment, this model here made it possible for us to conclude two things. The first one is that CTC clusters detach directly from the tumor as clusters, so as multicellular aggregates, and there is no aggregation of tumor cells in circulation. And the second is that when you look at the metastatic ability by means of this multicolor staining, we could determine that CTC clusters are 50 times more likely to form a metastasis compared to an equal number of single CTCs. So the experiments that I've shown you challenged the dogma of how metastasis happens. You probably have heard or have read publications about something called epithelial to mesenchymal transition. We were essentially showing the opposite of epithelial to mesenchymal transition. So we were saying that whenever cancer cells do not lose cell-cell junctions and retain their ability to form clusters, these cells are much more likely to form metastasis compared to cells that lose their cell-cell junction repertoire. However, when my lab moved to Basel, we literally had an open field in front of us. So we just discovered CTC clusters, but we knew nothing about their biology. So what really happens in a cluster that makes them so successful in forming metastasis, and most importantly, how can we target them? Can we develop any treatment for suppressing the metastatic ability of these clusters? So the way we started approaching this was quite ambitious from the start. So we aimed to do something technically quite difficult, which is a single cell resolution DNA methylation profiling of CTCs directly isolated from patient samples. As you know, DNA methylation is a epigenetic feature that happens in all cells, and that marks the DNA in very precise locations. And what generally it means is that whenever you have a methylation or a high methylation level in a certain portion of the DNA, the genes that are in this area are repressed, so there cannot be a translation of these genes into mRNA and then protein. And when there is a low methylation of the DNA in the corresponding area, there is, or generally, there is a higher expression of these genes. So we did that in CTCs, and we came across something very fundamental. So these are CTC clusters and single CTCs isolated from patients with breast cancer here in Basel and from matched within individual patients. And you can see that there are regions of the DNA of the clusters that are selectively hypomethylated, so very low methylation levels, and the very same regions of single CTCs are hypermethylated, so very high methylation levels. This was very clear. And we, of course, asked what type of regions they are. And this is a fairly complicated slide, but please just focus. I'm not sure if you can see my pointer. In case you cannot, just focus on panel B, the very first line. So this is the gene ontology analysis of the regions that are hypomethylated in CTC clusters. And essentially what we find is that these regions correspond to binding sites for three very important transcription factors. These are OCT4, SOX2, and NANOG. These are transcription factors that together are very powerful and can regulate stemness and proliferation of a variety of cell types, including cancer cells. So the idea here is that whenever cells have, cancer cells have cell-cell junctions active and can form clusters, they demethylate binding sites for stemness and proliferation transcription factors that can then do their job and confer these properties to the cancer cells. This slide is probably even more complicated, but the take-home message is very simple. So what we see here when looking at RNA is that the genes that are transcribed by OCT4, SOX2, and NANOG, they are also upregulated in a very coordinated fashion, all together, upregulated in human CTC clusters and downregulated in human single CTCs. So basically, cluster means stemness and proliferation. This was the finding. And we went one step further. We thought, okay, if just clustering can confer stemness and proliferation properties to cancer cells, what happens when we decluster? What happens if we separate these CTC clusters, the cells within CTC clusters, from each other? And that's what we did in here. So we used a library of FDA-approved compounds. So this was approximately 2,400 compounds approved by the FDA for treatment of many different types of diseases, not only cancer. And we screened these compounds for their ability to disrupt human CTC clusters isolated from patients. And we monitored at the same time viability. And you can see here the red dot is a control. So you can see CTC clusters growing without any treatment. So they have a certain size and they are highly proliferating. And you can also see that the vast majority of the drugs that we tested in this context didn't do anything to these clusters. So they didn't disrupt them, and they didn't kill them either. Then there was a set of drugs which decreased cluster size, but also decreased viability. And among these were many cancer drugs that are well known, but we were not really interested in those, because if they decrease cancer cluster size by killing, they would not really prove the concept that we had in mind. What we were interested on were the yellow guys that you see here. There were 39 compounds that consistently dissociated CTC clusters to the level of single cells. You can see the single cell control is the green dot. And at the same time, these cells maintained at least 70% viability, so still a high proliferation rate. So there was essentially no killing, but there was dissociation. And the interesting part was that many of the 39 compounds were targeting the same target, which is the sodium-potassium ATPase. We then moved on to mouse models, and we did the following experiment. We isolated CTCs from a patient with breast cancer. We transplanted them in immunocompromised mice. It always takes a while, so this is a 4-month-long experiment until the tumor reaches a certain size and the tumor starts to spontaneously shed CTCs and form metastases. And at this very point in time, we gave daily intraperitoneal injection of sodium-potassium ATPase inhibitors, in this case Wabane, and we kept treating for 3 weeks. And at the end of the experiment, we measured tumor size, CTCs, and metastases. So the result was quite exceptional, because we, with the 3 weeks treatment, we did not do anything to the tumor. So the tumor kept growing at a very high rate. So there was no difference in tumor size between untreated and treated mice. But if you look at CTCs, these tumors, the treated ones, started producing more single CTCs and less CTC clusters. So this is consistent with the dissociation of tumor cells from one another, or weakening of their cell-cell junctions. And simply this little imbalance in the proportion of CTC clusters and single CTCs resulted in an 80-fold, this is a log scale, so 80-fold decrease in spontaneous metastatic dissemination. And you can see the pictures here. So there were mice that had no metastasis at all. So to wrap up on this particular part, what I've shown you is that CTC clusters and single CTCs are very different from a DNA methylation standpoint, with clusters being hypomethylated in binding sites for OCT4, SOX2, and NANOG. By the way, this methylation signature, I haven't shown you the data, but it's on the paper, which is also freely accessible if you're interested in it. This methylation signature is also directly predictive of a poor prognosis in patients with breast cancer. But most importantly, we develop a new strategy to delay metastatic onset by treating with agents that can disrupt CTC clusters, and this in preclinical models leads to suppression of metastasis. I told you before that some CTC clusters come along with immune cells, and this area of research was also completely unknown when we started to get interested in it. We didn't know, for example, what type of white blood cells they were, and we didn't even know whether they were there to kill or to help CTC clusters. So we knew literally nothing about them. So in this project here, we investigated the CTC white blood cell clusters in multiple contexts. So in patients with breast cancers, in different breast cancer mouse models, some of them derived from xenografts with human cells. Some others were genetically engineered mouse models, and therefore were retaining a full immune system. And no matter what we were looking at, but we were seeing the CTC white blood cell clusters pretty much all the time. They come along at very different frequencies compared to single CTCs and CTC clusters without white blood cells. Sometimes they're very frequent, sometimes they're very rare. We do not yet understand what drives this. We can simply observe it. We do not know what dictates their frequency yet. But what is extremely clear is that whenever patients with breast cancer, and this is a cohort here in Basel, whenever patients with breast cancer have at least one of these CTC white blood cell clusters in their blood, they progress through their disease much faster compared to patients that don't have CTC white blood cell clusters. And among them is also patients that have CTCs of different types. What we did then was to ask what type of white blood cell is traveling together with the clusters. And to do that, we isolated CTC white blood cell clusters from the blood of women with breast cancer. We, with a robotic micromanipulator, we dissociated individual cells from each other, and then we did single cell RNA sequencing and compared the transcriptome of the cluster-associated white blood cells, which you see here in red, to that of reference white blood cells isolated from the very same patient. Then we looked at the data, and it was very clear from the beginning that cluster-associated white blood cells associate, at least for what concerns their transcriptome patterns, they associate very directly with myeloid cells. These can be monocytes or granulocytes. And then we repeated the whole experiment in mouse models, and we confirmed exactly the same, that the cluster-associated white blood cells, they have a transcriptome which is very similar to the myeloid cells of the mouse. And then with additional investigations, so looking at specific markers such as LY6G, we could determine with high precision that whenever you see a white blood cell associated to a CTC, 9 out of 10 times this white blood cell is a neutrophil, and 10% of the times is a monocyte. Then we did the opposite exercise. So we looked at the transcriptome of the CTCs that travel with the neutrophils and compared that to the CTCs that travel by themselves. And here the results were extremely clear. So the CTCs that travel with the neutrophils, they upregulate a set of genes that describes a very active cell cycle and DNA replication machinery. So essentially what they do, they proliferate much faster when they are attached to a neutrophil compared to when they are traveling by themselves. So it's like if the neutrophil is protecting them and is favoring proliferation and possibly metastatic ability, which I will show you in a minute. This is an interesting experiment. Here we demonstrated, I don't want to go too much into detail here, but we either with depletion of neutrophils through treatment with neutralizing antibodies for LY6G, or increase of neutrophil infiltration into the primary tumor by means of expression of GCSF, granulocyte colony stimulating factor, we could literally suppress or increase metastasis quite dramatically in mouse models. So just targeting neutrophils, so not touching at all tumor cells, just to show you how the importance of neutrophils in the metastatic spread of cancer. This is an experiment that took us almost 10 months to achieve, very technically challenging, but very important at the same time. So what we did here was to evaluate the direct metastatic ability of the 3 types of CTCs that we know: single CTCs, CTC cluster, and CTC-neutrophil cluster. And the way we did that, we first of all, we let tumors form in mice. And then once the tumors spontaneously shed these 3 types of CTCs, we did isolate them, we micromanipulated them, and then injected them in recipient mice, which were completely tumor-free, and measure metastatic development and survival of the injected mice. And we could determine that CTC-neutrophil clusters are the most metastasis-competent CTC subpopulation, followed by CTC clusters, and then single CTCs, which have very little ability metastasize. After doing all this, we realized that we were missing a very key information, which is how do CTC clusters form in the very first place. This was entirely unknown. There were theories that would argue that clusters are formed through a stochastic process of cancer cells falling into the bloodstream in a passive way. Many other theories around, but very little data on this. And we decided to tackle this part as well. And we looked at RNA sequencing data that we developed and realized that there might have been a strong connection between CTC clusters and hypoxia. And as you know, hypoxia is defined as either the lack of oxygen or let's say a reduced availability of oxygen in a cell. And to investigate that, we engineered our cells in a very particular way. So we engineered them with a construct that makes them turn yellow. So they start expressing yellow fluorescent protein whenever they experience hypoxia. And so we injected these cells in mice. We waited for tumor formation. And the literature says that hypoxic tumor cells are always located in the core of the tumor. So that could have been a scenario. But in reality, this experiment allowed us to test all possible scenarios, and also including those where hypoxia is less confined to the core, but it's more scattered across the tumor. And indeed, we observed many different outcomes. So this is, for example, a tumor where you see a very small hypoxic core in the middle. So this is the very early hypoxic core at its onset. And here you see a different tumor that has hypoxic regions scattered throughout the tumor itself, so not really confined to the core. What we could say, though, as a general rule, is that hypoxia occurs in regions of the tumor that are poorly vascularized. They do not have zero blood vessels. They do have blood vessels, but they have less blood vessels than the regions that are normoxic. And you can see it very well in the figure on the right side, where the blood vessels are labeled with the blue dye. And you can see them very much present in the middle where the tumor is normoxic, but then there are areas of hypoxia labeled in yellow where the amount of blood vessels is severely reduced. And so then what we did was to take these tumor cells, which were now labeled as a function of their hypoxic status, and we did a full proteomics analysis, so mass spectrometry of tumor cells directly isolated from tumor-bearing mice and labeled with this differential hypoxia labeling strategy. And the proteomics analysis told us something very interesting. I think if you, before this experiment, if you were to Google hypoxia and cancer, what you would probably find would be many papers that say that hypoxia leads to, again, epithelial to mesenchymal transition. If you look at these papers in detail, you would discover that most of these statements are derived from in vitro experiments, while there is very little, if not nothing, in vivo. And so when we did this experiment and did proteomics analysis of the cells, we found again exactly the opposite. We found that cells that experience hypoxia in the growing tumor, they also upregulate very strongly cell-cell adhesion molecules and cadherins to boost their cell-cell junction repertoire. And then as a consequence of that, you can imagine the rest. So as a consequence of that, we find that the vast majority of CTC clusters is hypoxic and has a very high cell-cell junction repertoire, while single CTCs most likely derive from normoxic tumor areas, and they have, of course, a low cell-cell junction repertoire. Then what we did also was to investigate the metastatic ability of CTCs as a function of their oxygenation. So we found that single CTCs, even when they are hypoxic, they are not good at forming metastases. So hypoxia doesn't help them much. And that whenever CTC clusters are hypoxic, they really can show their full metastatic propensity. The data that I showed you on hypoxia has some quite profound consequences, I think. So the main one is shown here. We did an experiment which can be quite provocative in nature. And the experiment is the following. So we took tumor-bearing mice and we treated them with bevacizumab, which is, as you know, anti-angiogenic therapy. So the target of bevacizumab is VEGF-A, and the suppression of VEGF-A leads to a reduction in the vasculature. As expected, when you give bevacizumab to tumor-bearing mice, and this data is out there in the literature since many, many years, tumors shrink. So they have less blood vessels and they are smaller. However, that's where everyone stops usually. What we did in this case is to look at this a little bit more in the context of metastasis. And what we found is that these tumors treated with bevacizumab, even though they are very much smaller, they are much more hypoxic. And the consequence of being a lot more hypoxic is that there is a much higher release of CTC clusters and a huge increase in metastasis. So these mice have very small tumor, very big metastasis, and of course they live less longer than mice that are not treated with bevacizumab. So this could also be one of the reasons why clinical trials with Avastin in patients with breast cancer have failed. And we think that one of the reasons could be that while tumor shrinking may occur, there is clearly an increased dissemination through CTC clusters. Then we tried to do exactly the opposite. So we tried to give more blood vessels to the tumors. In this case, we did that by treatment with Ephrin B2. Ephrin B2 is a protein that binds to its receptor, Ephrin B4, on the endothelial cells and promotes controlled proliferation of endothelial cells. And so you can achieve a higher blood vessel density by treating with Ephrin B2 and still having functional blood vessels. And so we did the experiment, and you can see something very striking here. So you can see that control mice in this particular tumor model, they live for about 6 months, and their tumor doesn't get too big, but it's already enough to shed CTCs and make metastases and kill the mouse. When we treat with Ephrin B2, the tumor gets a lot bigger because we are giving a lot more blood vessels to the tumor, so there is a lot more nutrient, more oxygen, so the tumor grows faster. Yet, what happens is that there is less hypoxia. And so you can see in the next pie chart that there are no CTC clusters released from these tumors. And as a consequence, these mice do not form metastases, or they form much less metastases compared to control mice. And of course, as you can already appreciate from the graph on the left, they can survive much longer, even though they have a bigger tumor. So to wrap this up, on the biology part, I've shown you that tumor cells can be released into blood of patients and of mouse models in different ways. They can be released as single circulating tumor cells. These are generally normoxic, and they generally do not have the capability, or they do not have a high capability to generate metastasis. Then tumors can also shed CTC clusters and CTC-neutrophil clusters, which are typically hypoxic, and they are typically the pioneers of the spread of cancer, of metastatic development. And of course, what's next based on these findings is to see whether we can target CTC clusters directly in patients. So we have started treating patients with sodium-potassium ATPase inhibitors, particularly digoxin, which is approved here in Switzerland and throughout Europe and in the US, particularly for treatment of patients with cardiac arrhythmias. And what we are doing here, we want to really demonstrate that the cluster dissociation, like we saw in our preclinical models, cluster dissociation is possible and as well as measurable also in patients. And we are giving digoxin daily up to a certain level that it needs to be reached in the serum. We treat only for 3 weeks, so this trial doesn't have any clinical endpoint. So we just look at CTC clusters. And we then enumerate number and size of CTC clusters pre and post treatment along with the pharmacokinetics. This is done at the University Hospital here in Basel by phenomenal clinicians and investigators. And we have started already treating patients, and we, despite COVID, we are well on track, and we are aiming for a completion at in the second half of 2021. Okay, so I'm having 2 minutes left. So take-home messages. I hope I could convince you that CTC clusters are very important, and they are precursors of metastasis in breast cancer. We think they are also precursors of metastasis in many other cancer types. Particularly, they are so metastatic because they can hypomethylate binding sites for OCT4, SOX2, and NANOG, as well as other stem cell and proliferation-related transcription factors. Also, we identified FDA-approved compounds that can dissociate CTC clusters and suppress metastasis in preclinical models, and we are now testing them in patients. And also that neutrophils can be yet another way how CTCs can boost their metastatic ability and increase proliferation. And finally, that intra-tumor hypoxia leads to cell-cell junction upregulation and intravasation of cluster CTCs with a high metastatic ability. Just as a quick note, my lab is going to move from Basel to Zurich, so not very far away, starting from January 1st, and we are, of course, looking forward to expand our studies to a lot more models and a lot more cancer types and a lot more exciting questions to be answered on CTCs. And last but not least, I think this is the most important slide of all. These are the people, the scientists that did the work that I told you about. So people in my lab here in Basel, a lot of fantastic clinical collaborators at our university hospital, colleagues in the bioinformatics and imaging course, colleagues in Zurich at the ETH, Germany, US. I forgot also to mention Angle for sponsoring this webinar. Thank you very much. And also thank you to the funding agencies for supporting our work. Thanks a lot.
Thank you, Professor Aceto. I'd like to welcome Paul Smith from Angle now, who will also be taking questions on the panel. So I'm just going to read out the first question. Okay, so looking at the CTC-neutrophil clusters, a question for Professor Aceto. What is the clinical monitoring implications of your finding?
Yes, so if I understand the question correctly, you are asking me how should it be implemented, monitoring of CTC neutrophil clusters in the clinic?
Yeah.
Yes. So my answer is that clearly a snapshot analysis is not sufficient. So I think patients should should be asked to provide blood samples progressively as their disease evolves and as they get new treatments, as they respond or as they relapse. And even when disease is apparently stable, I think the emergence of the CTC neutrophil clusters could occur pretty much at any time during disease progression. They could be there already early, they could arise late. So I would definitely recommend to analyze multiple blood samples and during disease progression.
Thank you. It's a lot of the questions are from practicing physicians to try to understand how it works in the translational oncology part, to say amazing findings, what does that mean for me in the clinic? So it's that kind of like, what's the next step? Next question from the audience is still based on the clusters. And how do the question for Paul? So how does the Parsortis system capture clusters in terms of why doesn't it break them up, you know, and why do they not break when they go through the actual filter?
Absolutely. So this is also a very important question. There is a chance, of course, that if you run CTCs through a narrowing gap, that shear forces might increase and the clusters might break. Of course, we measured that. This was one of our first worries when we started processing samples with Parsortis. On one hand side, you want to increase capture rate, so you want to make sure that you capture all your CTCs, so not only the clusters but also the single cells. On the other side, you want to make sure that you don't alter any of the CTCs that are isolated from a patient. And there are two scenarios. One is what Paul has mentioned, that you could break clusters, and the other one is the opposite, that you could generate artificial clusters on chip. And so, of course, we did control extensively for both scenarios. We developed in-house protocols that are dedicated exclusively to this. So we developed protocols that ensure a very high capture rate of CTCs without having to run into issues for cluster aggregation, artificial aggregation of CTCs or cluster disruption. And it has to do with a number of parameters that we had to improve, and that we keep monitoring. And of course, these parameters may change from cancer type to cancer type. So what we have developed is a very efficient protocol for breast cancer and also for prostate cancer. What we are starting to notice is that depending on the cancer type, the protocol should probably be slightly adapted, because the interactions between cells, between cancer cells and between cancer cells and immune cells, the strength of these interactions may change depending on which cancer type you're looking at. But it's the devil is in the details, so it's all about the details of the protocol. Absolutely.
Thank you, Professor Aceto. With regards to the details and not the devil, Paul, could you answer about the technology side in terms of, so the researcher was asking to say, looking at different systems, why does Parsortis not break it up, and like say, why does another system catch it or not catch it through EpCAM? If we're looking at the clusters and these have got like 50-fold more metastatic potential, then surely the technology for the filter side before we get down to the downregulation is quite critical.
Yeah, absolutely, and great question. And I do have to recognize the amount of work that Professor Aceto and his group have put into understanding this, because they also generated the tools and the models that mimic clusters in real life, and it's hard to optimize unless you have something that's representative. And clearly, if you look at the literature, techniques have been missing clusters for many years, and so that's part of the issue. I would say again, part of what Professor Aceto said. The devils are in the details. Parsortis is designed such that it can provide a fluidic environment to capture cells where the shear stresses that the cells see at any point can be kept below those that they would see in the circulation. And I think that is the key. And that is from a variety of points of view that contribute to that. But basically, our goal is to provide an environment where again, two types of forces are never exceeded. One is the shear stress, around a cluster or around a single cell, and that's part of the reason for using this gap approach, as well as the width of that terminal gap. And the second is actually also avoiding any situation where the sort of transmembrane pressure on an individual cell is not exceeded beyond what it would see in real life. And that can be a challenge, for instance, in sort of direct filtration approaches where effectively you get a cell over a hole, over a pore, and it's taking the entire pressure drop of whatever that filter is across the cell membrane. With regards to sort of epitope capture approaches, the challenge there is I think the heterogeneity of CTCs, which has again been illustrated here, both between disease, between stage, depending on treatment, etc. So taking sort of an epitope agnostic approach, is what sort of gives the bigger window. And the other approach of, sort of using targeted antibodies to deplete and sort of leave behind, now is also sort of brought into question because of the fact that, you have these neutrophils associated with clusters, and if you're doing a generic WBC kind of depletion, it's been shown that you lose those. Things as simple as centrifugation have been shown to break up some of these clusters. So it's all about, as I like to say, how we'd like our politicians about being gentle and inclusive.
Thank you. Next question that's coming is, stay on that basis. So Professor Aceto, you said about the artificial clusters. How are you telling the difference between them in terms of ruling out false positives?
Yes, so you typically run control experiments where you spike in healthy blood, for instance, only single cells. And you can even go to very high numbers. So you can spike thousands of single cancer cells, which you would never find in patients, but just to be in the worst case scenario. And then look at what you find on chip. If you still find all the single cells or if you find artificial aggregates. And that's how we typically measure the likelihood. So we do this all the time, and we measure the likelihood of our protocols to cause artificial aggregates, which are excluded.
Thank you. Next question, I've got another one from a practicing oncologist. So you spoke of the 39 compounds that are already FDA cleared, and then I think that you said that they were repurposed. Is there any literature out there? Is that the next step in terms of adding in these, I think it's the sodium potassium drugs to help for the cluster busters? So is that in the next clinical implication? Because I think the oncologists are keen to actually start going, we don't have to wait 10 years for a new drug if there's something already cleared now and it will help.
Yeah, I think I fully agree. I think the next step is to see whether what we have observed in preclinical models is true in patients as well. And that's why we are doing this very small clinical trial. It's only 12 patients. And the reason we are doing it this way is because we want to have the answer pretty fast. and we do not have any clinical endpoint. So we treat patients with sodium-potassium ATPase inhibitors to see whether we can truly dissociate clusters of CTCs in women with breast cancer the same way we did in our models. And then if that's the case, then by all means, I think we should accelerate on that.
Okay, because it's all to do with, you know, how fast can we do this for patient benefit? And then in terms of moving to the next stage for clinical application, it always comes with monitoring as well. Would that be a biomarker along with it, CDX, a clinical monitoring tool? If the oncologists are prescribing these drugs, how do they know if they're making it better or not? You know, what's the clinical part of it?
So that's a more complex answer. So I think we are still thinking about this in detail. So first of all, we need to give some thought about what type of patients could benefit from such a therapy. You could argue that certain cancers spread so quickly that it's hard that even if you have the cluster buster therapy as you defined it, maybe you're already too late. In other cases, disease spreads so slowly. We know some breast cancers take decades to metastasize, or at least metastases become clinically visible decades after the primary tumor. And it's unlikely to have a clinical trial that is lasting so long. So first of all is which patient would benefit. We are looking into this in great detail at the moment. I think it's not everyone. I think it's only a proportion of patients that clearly have CTC clusters in their blood. So their disease spreads through CTC clusters. And secondly, that have the disease dynamic that would make a cluster treatment eligible. That means that their disease still needs to spread, or that their disease spreads in a different way from what you know, from what you could imagine, because much of the literature, and we could discuss this for a very long time, much of the literature argues that cancers, or most of the cancers, spread early. And that all the metastases are seeded from the primary tumor very early on. This is not exactly the case, I think, because if you look at very, very good literature that came out in the past 4 or 5 years, when people did finally tumor metastasis phylogeny studies and they sequenced different metastases from patients, they realized that there is a great extent of metastasis to metastasis dissemination rather than primary to metastasis. And then in those patients, I think it would make a lot of sense to have a cluster buster approach, because you would delay the seeding of further metastasis, even if the disease is already at stage 4.
I understand. Second question came in after this is within the different cancer tumor types, have you seen the CTC clusters more apparent in some types than others, or?
Yes and no. So we do see generally more clusters in people that have, in women that have hormone receptor positive breast cancer.
Okay.
Yet we see them when they have bone metastasis already. So it's already a disease which, you know, you could argue whether it is still hormone dependent or not. I'm sure you're familiar with the literature there. Disease switching after treatment, and then especially at the metastatic stage. So I don't know if it's the hormone dependency that helps, or if it's the metastatic site that helps, or if it's just the fact that there is more disease, and then all of a sudden we see more cells in circulation, and then we see that there are more clusters. But so typically it's those patients. It's women that have a hormone receptor positive breast cancer,
okay,
that has already metastasized, and that is progressing quite rapidly.
How about androgen-driven prostate?
Yes, it's very similar. It's very similar. So patients with prostate cancer, they also have quite frequently bone metastases, and there we have pretty similar scenario as what we see with the women with metastatic hormone receptor positive breast cancer. So we do see a lot of CTCs, and we do see CTC clusters.
Can imagine with a varying mass as well. So next question is coming through. Looking at CTC clusters again on the hypermethylation for the OCT4, SOX2, and NANOG. Any kind of targets with which we can look at those transcription factors there? Is there any intervention that you've looked at to stop the transcription before it starts to go through?
Very difficult. We've looked at that. Of course, we could not find anything that could stop the transcription. So these are very, very, very fundamental transcription factors. So they are very conserved in all cells. Okay. Difficult way. I don't think this is feasible.
Okay. Next one that's coming is back to biophysical. So looking at the CTC-neutrophils, Paul, does the Parsortis look at capturing neutrophils, and does it vary in sizes, or how does it work?
Yeah, absolutely. So obviously, if these neutrophils are associated with a cluster, and they will be, the goal is to maintain that cluster integrity, and they'll be captured with that. But on the other hand, you know, there is a subset of cells, including neutrophils, you know, that can have the sizes and deformability to be retained along with the CTCs or other rare cells. And the you know, the harvest is not perfectly pure. We get a certain number of cells. It turns out that maybe that's a good thing. We've all worked so hard to remove everything, and you know, maybe we should spend more time looking at some of those specifically retained cells. And I know some of that work is going on, but yeah, it'll depend on their size. So you know, we've seen other types of you know, non-cancer related cells, for instance, like megakaryocytes, that are retained and have interesting properties relative to cancer in the harvest.
Thank you, Paul. So next one's coming through.
While you're doing that, can I just ask a quick follow-up question, if you don't mind?
Sure, please do.
Back to the cell junction inhibitors, I'm just wondering if you think the impact you see is happening sort of during you know, the formation or the growth of these cells in the hypoxic environment, or is it happening on the clusters once they're in the circulation, or can you?
Yes, I think almost nothing happens during circulation. I think circulation time is so short. So whenever clusters get released, or even single CTCs get released from a cancerous lesion, they circulate for a very short time. We measured this in mice. Obviously, we cannot do it in humans. At least we couldn't find a way to do it in humans yet. But in mice, they circulate for a couple of minutes, and then they get stuck somewhere, typically in capillaries or extravasate. So I think pretty much nothing happens in circulation. So in circulation is purely struggle for survival. There is a high shear. It's like jumping into a very, very rough river and just trying to hang on something. And it's all very quick. So I think all what we see in terms of biology, it has happened already at the level of the primary tumor. And the same way is for the drug treatment. So treatment with the sodium potassium ATPase inhibitor results in a dissociation of tumor cells or a weakening of cell-cell junctions in tumor cells already in the primary tumor. So when they jump into the circulation, they jump in as single cells.
Perfect. And the second part of that question, I'm just curious, because many of these drugs have been out there and used very widely, would you expect if it was feasible to do a meta-analysis of, for instance, cancer patients that have also been on digoxin? Would you expect?
We did that. We did that already. So that was one of the first things we did when we discovered the digoxin effect. Unfortunately, you find all sorts of things. I was not happy about the results of this meta-analysis. They were very confusing. So you could see patients in whom there seemed to be a very clear benefit, and other patients where nothing really happened. So it was very, very unclear. So yeah, I think we just have to do it prospectively.
Yeah, thank you.
Next question will come in and said that you developed a protocol to achieve a high yield of CTC capture, but you didn't talk much about the actual protocol. Could you expand just a little on the protocol?
Yeah, it's of course, it's a mix of things. It's a mix of factors. It's based on optimized flow rates to ensure high capture rate and to avoid disruption, as we discussed before. It removes quite a lot of the steps that are typically used for Parsortis. It adds additional steps which are done to make sure that there is no loss. Basically that's it. It's nothing special. It's just an optimized protocol for making sure that capture rate and preservation of the properties of CTCs are maintained at best.
Thank you. Next question that's came in is says, I'm wondering in the complex process of metastasization how extra vesicles fit in this model of metastasization induced by hypoxia through the cluster formation.
Yeah, it's a good question. I don't know. I think the straight answer is I don't know. There's not much we have done on EVs. I mean, I'm aware of the literature, of course. They seem to play a role in metastasis, at least in some models, but we don't know how they relate to CTC clusters.
Okay. Thank you. So next question that's came in is, Paul, you touched on EPCAM earlier. Is there a difference in terms of the different capture methodology and then what's available to downstream analyze after if things are missed out? So we've talked a lot about harvesting. Is there any differences between the different techs? And then what does that mean for later on looking at it? Because I think Professor Aceto looked at the downstream genomics as well.
Yeah, I mean, I think it's a great question because obviously there's a lot of things, many of which were demonstrated today that you may want to do with CTCs. So I would say the critical factor goes back again to ideally being able to harvest these with as little intervention as possible, so as viable and natural state as possible. And that's not just about sort of physical stress. And in the case of Parsortis, it's a matter of sort of gently back flushing the cassette to capture the cells in a fluid environment. But it's also other interventions, such as antibodies themselves, binding, particularly to if you're using sites to capture them that are involved in some of these mechanisms. Obviously, you're changing those cells when you're touching them. So I think the key is gentle, and then they are amenable for the sort of picking and separation technologies that have been illustrated here to allow them, understanding either the neutrophils, individual CTCs, or CTCs of particular cluster types. We have, and many other folks have developed specific staining approaches that allow identification of the phenotype of the cells as well as enumeration of clusters. And then again, we've developed some specific gene expression technology that allows us to look just basically in the entire harvest together to look at targeted, 100 or so targets at the same time. And one of the reasons we took that approach goes back to the original answer, is we basically harvest right into a lysis buffer, so we get a snapshot of the gene expression of those cells immediately as they are harvested. So because things change with time.
Thank you, Paul. So next question that's came in is, without breaking any confidentiality, Professor Aceto, what's next for you? So what's on the horizon that you're looking at that everyone needs to look out for from you?
Yeah, so we are clearly expanding to different cancer types. So you've seen my presentation was mostly focused on breast cancer. That's where we are strong, where we know already a lot about the biology. There are a lot of exciting questions in many other cancer types that are big killers, like melanoma, like pancreatic cancer, also prostate cancer, lung cancer. So we are really expanding to those, trying to understand more about how CTCs function in these different cancer types. I think it would be a mistake to assume that they function always the same way, no matter what cancer type. I think there might be very different phenomena going on. For instance, which partner cells they choose. So in breast cancer, they seem to prefer neutrophils, but I think in other cancer types, they may well go with some other, or potentially go with some other immune cell or stromal cell. So this is clearly one thing. So we're expanding to other cancer types. We are getting more and more into clinical studies, trying to translate what we find in the lab, because I think that's the goal. We tend to get quite bored with the lab at some point, and I mean, papers are great, but I think it's more important to translate our results, or to challenge our results in the clinical setting. I think that's what we are here for. And then clearly general questions on genetics and biology of circulating tumor cells. I think there's really a lot of unknown, even very basic things that might determine the outcome of a circulating tumor cells, whether it ends up metastasizing or not, and where, to which organ, for example. So these are all things we are looking at.
Thank you. You also mentioned about the translational part, and then earlier you spoke about the anti-VEGF drugs, where the vast majority of, they looked at to say, oh great, we shrunk the tumor, so we met the endpoint, but then people didn't look further on. So looking at that, and I believe that you said, tell me if I'm wrong, is that the anti-VEGF increased the hypoxia, and then the hypoxia increased more CTCs. What would be kind of like a small study, would it be worth studying in patients, in monitoring that, to say what's the real life situation? Because I think you mentioned about in vitro studies earlier. Would it be possible to look at patients with that?
I mean, I think Avastin is very well characterized already for the effects that can have on patients. I mean, there are certain tumor types where Avastin is used because it clearly provides a clinical benefit. What I was referring to is breast cancer, where Avastin is not really used because the trials didn't look so good. And I was mentioning that one reason could have been that certain tumor cells upon treatment with Avastin became more hypoxic. The literature on Avastin is quite complex. Also, I think the answer could go for long.
Yeah.
But one point which I would like to make is that if you look carefully, there seemed to be also a vascular normalization effect of Avastin. So not only generating hypoxia, but in certain tumor entities, what people have observed, and probably these are the tumors which have an exaggerated number of blood vessels, that if you treat with Avastin, you actually improve oxygenation of the tumor because you normalize the vasculature. So I think it's a complex discussion. It depends a little bit on the tumor entity. It depends also on the features of the specific tumor you're treating.
Okay.
How much vessels and how much perfusion the tumor has, concentration of Avastin, and so on, so on, so on.
I think it sounds like it's a more clinical monitoring for oncologists to look at, is to say, okay, we'll do this, but then we need to monitor it, the situation as well, which is where all the useful data comes in from the translational oncology to say, this could happen, so you need to monitor it, and then we can adjust it from there.
Sure.
That makes sense.
Exactly.
Okay, you spoke earlier about the future things that you're going to do. As we've got 200 oncology researchers and oncologists from around the world. Any collaborations that you're seeking that someone on the call might want to offer you at the moment to go, ooh, you're looking for this? So you've got a whole audience around the world. Any top collaborations you're looking for right now that someone might be on the call?
Well, we are quite well equipped in terms of samples in general. So we do get quite a lot of samples from many collaborators, not only in Basel, but also in Europe and also in the US. Yet, of course, it's always nice to increase the number of samples. I'm referring particularly to tumor types that are not so frequent. I think that this would be interesting to explore. There, the number of samples, of course, is lower compared to others, at least for our lab in this moment. So there would be nice to collaborate, and I think even more important than that, I think if there is any clinical question, that's usually how many of the projects in my lab start. We start with discussing with clinicians, and we ask them, what are the major issues that they are facing when they treat a cancer patient, and then we go from there. For instance, I can make an example. I mean, the whole project about hypoxia and CTC clusters started because we realized in a meeting with clinicians that we had no idea which patient would be positive for a cluster or negative for clusters before running the sample. And you could say, well, after you run a lot of patients, you probably have some features of the disease that may tell you what's the likelihood to see a cluster. And one of the questions before was also alluding to that. In reality, we don't have such a strong data, so we still are unsure of which patient might have a cluster and which one would not have a cluster. And that's how it started, because we realized we don't know how the clusters are generated in the first place. So then we started thinking about all the rest.
We've just had a lot of comments coming in saying, could you please introduce me, because we want to work together. So I think, yeah,
Great.
Now got a few people contact you with different samples. So Paul, looking at from an angle perspective, in terms of like getting into clinical studies, and it's all the monitoring, any kind of any work that's going forward or that could be gone forward from an angle perspective that you may want to speak to anyone out there to collaborate, because we've got a wide variety of oncologists and researchers and pharma on the call as well. So anyone particular out there that we might be looking at?
Yeah, no, obviously we're keenly interested in this area, and it's part of of our strategy, part of the reason that we've invested so much. And folks might have seen, I guess about 3 weeks ago now, we submitted to the FDA for approval of the Parsortis instrument for isolation of cells for further analysis in metastatic breast cancer specifically. And hopefully as they review, that'll be successful. And so in that case, it would be the first platform approved for FDA approved for isolation of cells. And so we're very interested in working with folks to sort of build out the knowledge base of not only what the cells and the clusters can do to answer specific clinical questions, But importantly, as noted earlier, is to ensure that we can, optimize the Parsortis protocols and operation for individual cancers. These are, extremely heterogeneous, and it's like any platform, it's designed to sort of operate within a range of parameters, and they may be slightly different for each cancer. But we're making an investment to make this tool certainly available, as a regulated device to assist in this work. And yeah, would love to hear from folks who have particular questions that might be relevant here. So.
Thank you, Paul. Professor Aceto, we've got a fair amount of clinicians on the call today, and a lot of people are keen for maybe a follow-up in the future, or or some more information on these 39 compounds, because I think a lot of people are thinking about the patients that they know now, whereas a lot of the research translates in 10 years' time, whereas there seems to be quite a keenness now, which is completely understandable, to go, help us with the information now. Because if there is drugs which are already cleared, and as oncology is an art, they can choose to see whether it might help or might not, because if it's cleared, they've got prescription jurisdiction. Sure. So, anything that could come out of your lab for that we could share with everyone, or maybe do a follow-up panel session or anything like that.
Sure, of course, we can discuss this further. What I can mention is that all the compounds that we screened have been published. They are available. The paper is open access now. So that's the Cell paper in 2019, and there you can clearly see which compounds worked, which ones did not. What I would say, though, is that the way we see it, and I'm a little bit skeptical always, because I've seen so many results being great in the mouse and failing in the clinic. And what I would like to say is that that's the very reason why we are doing this mini trial on 12 patients. We really want to understand whether these results could have a very meaningful outcome. And it's a very simple trial. It's very fast, and that's what we already have some results, of course, and they are extremely encouraging. Yet we need to wait and see and run the statistics at the end. But I think it's important not only to review the drugs that are on the paper, but also to test them prospectively in patients in a very controlled way, if that's the goal.
I think there's a keenness from the audience to see where they can support for the here and the now as well, because 10 years, is a tremendous amount of time within oncology for a lot of it. So there's a definite appetite to support you on all that. Looking at the time now, I think it's time to wrap up. So any final comments, any advice for the audience, Professor Aceto or Paul? If not, we can wrap up. Thank everyone for listening, and thank Angle for sponsoring this exceptional webinar delivered by Professor Aceto.
Yeah, so thank you very much. It was my pleasure to be here and to give you an update on our research. I hope this will be also an occasion to start new collaborations. So thank you very much for sharing this platform and to start the connections. This is great. Advice to the audience: I don't think I have any, apart from the one that if you're interested in CTC biology, it's a very exciting field, and it's a very challenging one at the same time. Be careful with the literature out there. There's a lot of papers which are not high enough quality and may be misleading. I think the whole field of EMT is a story which has been going on for a while, but then as soon as you look at patient samples, you don't see much of EMT going on. So I think that there's still a lot of questions to be answered, and I'm really looking forward to do that in the future.
Thank you, Professor Aceto. Paul?
Yeah, again, let me just thank you, Professor Aceto, for doing this. It's extremely exciting work, and obviously we wish you the best in pursuing this, not only in breast and prostate cancers, but beyond. And I think most exciting because, it sort of, at least illuminates the potential for, another approach, a completely different tool to deal with potentially more living with cancer as opposed to trying to eliminate cancer. So very exciting. And for, companies like ourselves, and I've been in the tools business, if you will, for many, many years, it's always gratifying to see people take a tool and do something so exciting with it. So congratulations, and I hope this has stimulated interest among the audience to also start thinking about things in this way. So thank you again, and thanks for all that have attended.
Thank you, Paul. Thank you, Professor Aceto.
Thank you, Paul.
To all, the webinar will be translated into a video and available in about a week's time, with which we will send out a copy to everyone. So if you did miss anything on the detailed transcription factors, you can watch it again with a coffee. Thank you, everyone. This webinar will finish in 3 seconds. Thank you, everyone.
Thank you.
Thank you.